Hello friends. So you have already studied a very good topic, an important topic on nitrogen metabolism from your botany paper called plant physiology. So today we'll discuss some practice questions on this topic, nitrogen metabolism, MCQs, multiple choice questions for your examination preparation, final examination that is going to be held for this semester. So we'll quickly begin. Okay, as you can see here, this picture representing the nitrogen metabolism or nitrogen cycle. And as you know, we usually start from here, nitrogen, which is present in the atmosphere in the form of molecular nitrogen, dinitrogen, which consists of two nitrogen atoms held together very tightly with the help of triple bond, covalent bond. And these bonds are so strong, they require such a large, tremendous amount of energy to break themselves, to separate two nitrogen atoms from each other, so that these two separated nitrogen atoms can combine with other atoms like hydrogen atoms and your hydrogen and oxygen atoms to form different compounds, and that process is called as nitrogen fixation. So that is happening naturally, as you have studied in case of lightning and thunder, where tremendous amount of energy is created. It is also being carried out industrially, artificially by Haber's process, where nitrogen is fixed. But the most important natural biological nitrogen fixation is carried out in which nitrogen is reduced into ammonia. So that is nitrogen fixation. That is the first step here. Then this ammonia is converted into nitrate, and that is oxidation of ammonia into nitrite first, and then oxidation of nitrite into nitrate. And this two-step process is called as nitrification. Formation of nitrate from ammonia is called as nitrification. It occurs in two steps: ammonia to nitrite. It is carried out by some certain microorganisms, certain bacteria, mainly nitrosomonas, and oxidation of nitrite to nitrate takes place by another group of bacteria called as mainly nitrobacter. So once nitrate is formed, it enters the plant because nitrate is the form that is mostly absorbed by plant, and after it is absorbed, it is again converted into ammonia. Now the question is, if ammonia is already available, why plants cannot accept, or why plants cannot absorb ammonia directly? As you know, ammonia, once it is absorbed inside the plant body, and if it gets accumulated, it will be toxic to the plants. So better they accept nitrate, they accept nitrogen in the form of nitrate, and then this nitrate is again converted, or it is reduced to nitrite and then to ammonia. And this ammonia now, either it is coming from here or it is coming from nitrate. That ammonia is used in the formation of amino acid, and that process is called as nitrogen assimilation. So that you have studied in amination process, two types of amination: reductive amination and transamination. Once that amino acids are formed, which are the building blocks of proteins, so proteins are formed inside the plant body, and in the food chain, those proteins get transferred to next trophic levels in the animals also. So after that, there is the death and decay of these plants and animals, so that this organic nitrogen containing amino acids, proteins, these are broken down to form ammonia again, and that process is called as ammonification. It is again carried out by certain groups of bacteria called as ammonifying bacteria. So what we discussed here, the first process, nitrogen fixation, which is a reduction of nitrogen to ammonia, then oxidation of this ammonia to nitrate, that is called as nitrification. That is the second process. Then we studied nitrate is converted into ammonia, and this ammonia, that is a reduction process. Nitrate is converted into nitrite and nitrite into ammonia inside the plant body, and then ammonia is assimilated inside the plant. That is called as nitrogen assimilation. And after death and decay, or when there is excretion from the animals, that means excretory or waste products, which contain organic nitrogen, and then from decomposition or decay, the nitrogen is again freed in the form of ammonium. That process is called as ammonification. And this is happening when oxygen is available. So these are aerobic processes. Nitrification is aerobic. These bacteria are aerobic. Nitrifying bacteria are aerobic. They are doing aerobic respiration. Whereas under anaerobic condition, the nitrate is converted into nitrite, nitrate into nitrogen oxides here as shown. And ultimately the final product is atmospheric nitrogen. So conversion of nitrate to nitrogen, this is denitrification process. This is a reduction of nitrate to nitrite and nitrite to nitrogen. So this is denitrification carried out by another group of bacteria called as denitrifying bacteria. Then one more process is there in which there is the direct oxidation of ammonia with the help of NO2 here, and that process which forms nitrogen, it is called as anammox. So with the help of these processes, nitrogen fixation, nitrification, nitrogen assimilation, ammonification, denitrification, and anammox, we are going to discuss some multiple choice questions on this nitrogen metabolism. So let's begin with that. We'll take the first question. Okay, nitrogen is necessary for the synthesis of what? Okay, no, nitrogen is necessary for the synthesis of proteins, compounds containing carbon, hydrogen, oxygen, and nitrogen. So B is the answer. Second, nitrogen is an essential component of what? Proteins, DNA, chlorophyll. You have studied this. It is an essential component of all of these proteins. Nitrogen is present in amino acids, building blocks of proteins. It is present in nucleotides, building blocks of DNA and RNA. It is present in chlorophyll. It is present in ATP. So all these compounds or all these substances require nitrogen. So option D is right, all of these. Next one, which of the following secondary metabolites contain nitrogen? Tannins, terpenoids, alkaloids, phenolics. Here the answer is alkaloids. Alkaloids are nitrogen containing secondary metabolites which contain nitrogen atom in a heterocyclic ring system. So C is the answer here. Okay. Next one, next question. The percentage of nitrogen in the air is about, okay, you know, the percentage of nitrogen is highest in the air. It is about 78%. Some say it is 79%, but the highest percentage is of nitrogen. 21% is oxygen, 0.03% is carbon dioxide. So here answer is B, 78%. And you know, the nitrogen is present in such a large amount, it cannot be easily broken. It cannot be easily fixed. It cannot be fixed by all the plants. It is the only ability of certain prokaryotes or microorganisms, and these organisms are called as biological nitrogen fixers or diazotrophs. Okay. Okay. The process of conversion of nitrogen to ammonia or nitrogenous compounds is called as, just now we discussed, that process is called as nitrogen fixation. Nitrogen is converted into ammonia, and the organisms are called as diazotrophs or nitrogen fixers. A is the answer. The formation of organic nitrogen compounds like amino acids from inorganic nitrogen compounds like your ammonia or ammonium ions, and that process is called as nitrogen assimilation. Just now we have studied that. Okay, D is the answer. The oxidation, biological oxidation of ammonia to nitrite and then nitrite to nitrate. This is two-step process carried out by nitrifying bacteria, and this process is called as nitrification. Conversion of or oxidation of ammonia to nitrate through the intermediate nitrite. So this is the answer. B is the answer. Okay. The reduction of nitrate and nitrite to gaseous nitrogen through a series of intermediate gases, nitrogen oxide products like nitrous oxide, just now we studied, that process is called as denitrification. So that is reduction process. Reduction of nitrate to nitrogen is called as denitrification, carried out by denitrifying bacteria. Denitrifying bacteria are anaerobic bacteria. Nitrifying bacteria are aerobic bacteria. So here C is the answer. Plants absorb nitrogen from the soil mostly in the form of nitrites, nitrates, ammonia, nitrogen gas. Okay, mostly they absorb nitrates here in the form of nitrates. They can also absorb nitrates and ammonia, but nitrates is the most easily absorbed form. Just now we studied. Once nitrates are absorbed, they are again converted into your nitrites and ammonia. So ammonia is then utilized for nitrogen assimilation. Okay. B is the answer. During nitrogen fixation, the enzyme nitrogenase adds how many hydrogen atoms to each nitrogen atom? You know, there are 3 nitrogen atoms joined together by 3 covalent bonds. So you have studied during nitrogen fixation, this nitrogenase enzyme complex adds how many hydrogen atoms? 3 hydrogen atoms to each nitrogen atom, because valency of nitrogen is 3. So NH3 is formed. First what forms? NH, double bond NH, then NH2, single bond NH2, and then there is formation of 2 separate molecules of NH3 from a single molecule of nitrogen, which is a dinitrogen. 2 molecules of ammonia are formed, and therefore here the answer is 3. Okay. The microorganisms capable of biological nitrogen fixation are called as what? Okay. Autotrophs, saprotrophs, diazotrophs, autotrophs. Just now I told you such microorganisms are called as diazotrophs. Dia means 2, azo means nitrogen, trophs means nutrition. Those microorganisms or prokaryotes whose nutrition is nitrogen, whose source of nutrition is nitrogen, are called as diazotrophs, nitrogen fixing organisms. C is the answer. Okay, matching type question. You have to match column 1 with column 2 regarding the free living nitrogen fixing organisms. So you know there are 2 types of nitrogen fixing organisms, symbiotic and non-symbiotic. Non-symbiotic organisms are called as free living. They live freely in the soil without any association with any other higher plants. So column 1 is given. Sorry, aerobic photosynthetic, non-photosynthetic, anaerobic photosynthetic, and non-photosynthetic. In column 2, their examples are given. So you have to match, and accordingly you will get the answer. Aerobic photosynthetic, Nostoc anabina. So these are blue-green algae, cyanobacteria. Aerobic non-photosynthetic. Okay, here example is Azotobacter, Pseudomonas. They are aerobic, but they are non-photosynthetic. They are chemosynthetic. They are chemotrophic. They are autotrophic, but they do chemosynthesis, not photosynthesis. Anaerobic photosynthetic are Chromatium, Chlorobium. These are, colored bacteria, colored photosynthetic bacteria. They are anaerobic, but they are photosynthetic. Chromatium is your purple sulfur bacterium. Chlorobium is green sulfur bacterium. There is one more bacterium which is non-sulfur, purple non-sulfur bacterium called as Rhodospirillum. That is also anaerobic photosynthetic. And anaerobic non-photosynthetic or heterotrophic is Clostridium. So after joining this, you will get the answer, proper answer. Option D is the answer. Okay, which of the following is symbiotic, aerobic, non-photosynthetic nitrogen fixing bacterium? So options are given. So you know symbiotic, Rhizobium is having symbiotic association with the root nodules of leguminous plants, members of family Leguminosae, now called as Fabaceae. So symbiotic, they are aerobic, they are non-photosynthetic, and that is the reason why they live in symbiotic association with the root nodules for the supply of food material. They are aerobic, and as they are aerobic, you have studied their nitrogenase enzyme is sensitive to oxygen, and that is the reason why that nitrogenase has to be protected. And there is you have studied there is one leg hemoglobin protein is there, which acts as oxygen scavenger, and it protects that your nitrogenase from more amount of oxygen. So here answer is Rhizobium, which is symbiotic, aerobic, non-photosynthetic, nitrogen fixing bacterium. Okay. Next one. Rhizobium is what type of bacterium? Yes, now we studied it is aerobic and it is gram-negative type of bacterium. D is the answer. For conversion of one molecule of dinitrogen, because it consists of 2 nitrogen atoms, when it is converted into 2 ammonia during nitrogen fixation by enzyme nitrogenase, it requires how many molecules of ATPs? So you have studied that nitrogenase enzyme complex requires 8 electrons and 16 ATPs to convert dinitrogen into 2 ammonia molecules. So B is the answer here. Nitrogen fixing cyanobacterium called nostoc is not associated with which of the following plants? That is the question. So you know, nostoc is associated with Anthoceros bryophyte. It is associated with Macrozymia or cycads, gymnosperms. Nostoc is also found in Gunnera plant, that is a member of Angiosperm, but it is not found in Azolla. In Azolla, which is a member of Pteridophyta, water 4, there is another blue-green algae and it is called as Anabina. So B here is the answer. Which of the following water fern is used as a green manure in rice fields, fertilizer in rice field? So Azolla is the answer. It is used as green manure because it consists of Anabina, which helps in nitrogen fixation for this water fern. Water fern Azolla for nitrogen fixation shows symbiotic association with which cyanobacterium? Just now we discussed that. So that cyanobacterium is called as Anabina. Anabina Azolla association is famous. B is the answer. Okay, symbiotic nitrogen fixing organisms. You have to match 2 columns. Symbiotic nitrogen fixing organisms. Column 2 is also given. Their examples and options are given. Now you know Rhizobium, symbiotic legume plants, leguminous plants, members of Leguminosea fabaceae. Frankia, these are actinomyces. These are included in bacteria, but they are filamentous structure, which are regarded as the organisms which are intermediate between bacteria and fungi. So Frankia is a member of actinomyces. It lives in symbiotic association with non-legume plants like Myrica, Ulmus, and Casuarina. These are called as actinorhiza plants. So B is matched with 3: Rhizobium, legume; Frankia, non-legume; Anabina, just now studied. Anabina is a cyanobacterium blue-green algae. It lives in a symbiotic association with Azolla plant. Okay. Klebsiella, it lives in a plant like Dioscorea, your highly medicinal plant. It forms leaf nodules in Dioscorea. So Klebsiella, leaf nodules. So after correct matching, you will get the right answer, and that is option C is the answer here. Which of the following bacterium is found in root nodules of soybean plant, which is a member of Fabaceae, Leguminosae family? Now, in most of the Leguminosae, you know, there is association of Rhizobium, but in case of soybean plant, instead of Rhizobium, there is another member of bacteria that is called as Bradyrhizobium. So D is the answer here. Which of the following is the source of nitrogen for plants? Okay, source of nitrogen for plants is not air. Atmospheric nitrogen is not available for plants. Plants cannot fix atmospheric nitrogen. That nitrogen has to be fixed by prokaryotes or microorganisms. You have studied that legume plants get it directly in the form of ammonia from your rhizobium, and other plants get it from their soil, and soil is the source of nitrogen. Nitrates are present in the soil. So C is the answer here. Which of the following is or are free living aerobic nitrogen fixing bacteria? So free living aerobic nitrogen, free living means they are not in any symbiotic association. They are aerobic, requiring oxygen for their respiration, and they are nitrogen fixing bacteria. And the answer is Azotobacter and Bajerinkia, named after the scientist Bajering, who is regarded as father of virology and also credited with the discovery of nitrogen fixation. So here answer is A. Which of the following bacteria are responsible for reducing the soil fertility? So denitrifying bacteria reduce soil fertility. Other bacteria increase soil fertility. Nitrifying bacteria or aminofying bacteria or nitrogen fixing bacteria, they increase soil fertility, but denitrifying bacteria reduce the fertility of soil. So here answer is option B. Which of the following bacteria are anaerobic, not requiring oxygen for respiration? Denitrifying bacteria are anaerobic bacteria. Nitrifying bacteria are aerobic bacteria. So answer here is option B. The process of conversion of proteins, nitrogen containing substances, into ammonia, that is conversion of amino acids into ammonia or ammonium salts by soil bacteria, is called as Okay, we have said that the process is called as ammonification. Formation of ammonia is ammonification. D is the answer. Okay, again matching type question. Column 1, column 2 is given, and you have to match the two columns exactly. Okay, nitrogen fixation is nitrogen to conversion of nitrogen to ammonia. Ammonia to nitrate conversion is nitrification. Ammonia to amino acid formation is called as nitrogen assimilation or amination. And amino acids are converted to ammonia during the decomposition or decay process, that is called as ammonification. So all options are correctly matched. A, B, C, D, 1, 2, 3, 4. So here the correct option is, answer is option A. Okay, the share of biological nitrogen fixation in the overall nitrogen fixation. Biological nitrogen fixation is the main process of nitrogen fixation, and its share is 90% of the total nitrogen fixation. The other being physical nitrogen fixation or industrial nitrogen fixation, they are having very less share. So C is the answer here. The oxidation of ammonia to ammonium or ammonium to nitrite. We have studied this conversion of ammonia to nitrite. This is first step of nitrification. It is catalyzed by what? Okay, the answer is very simple. This is oxidation of ammonia. Ammonia is oxidized to nitrite. So this is carried out by bacteria, and these bacteria are called as ammonia oxidizing bacteria. AOB, ammonia oxidizing bacteria. We have studied this, ammonia to nitrite. For example, nitrosomonas. Nitrosomonas is AOB, ammonia oxidizing bacterium, AOB. Okay. Subsequently, the oxidation of nitrite to nitrate is catalyzed by. Now you can answer this very simple. Nitrite to nitrate. Nitrite oxidizing bacteria, NOB. Nitrite oxidizing bacteria. And the example is nitrobacter. B is the answer. Nitrifying bacteria. Conversion of ammonia to nitrite and then nitrite to nitrate is carried out by, yes, you have to remember that sequence. First step is ammonia to nitrite. It is done by nitrosomonas, and nitrite to nitrate is done by nitrobacter, NOB. You can remember here NOB, NB, nitrobacter, NOB, and this is AOB, ammonia oxidizing bacterium, nitrosomonas, and nitrite oxidizing bacterium, nitrobacter. So here B is the answer. Okay, oxidation of ammonia to nitrite is not catalyzed. So oxidation ammonia to nitrite. So ammonia. So question is, which of the following is not ammonia oxidizing bacterium? Ammonia oxidizing bacterium. So nitrobacter is not AOB, it is NOB. So answer is D. So this is googly type question. So nitrosomonas, nitrosospira, and nitrosococcus all belong to one category. Nitrosomonas, nitrosospira. You can see here the word so, nitroso, and here nitroso. Nitrosomonas, nitrosospira, nitrosococcus. They are all belonging to one category. Nitrobacter is different. Okay, oxidation of nitrite to nitrate is not catalyzed by. So here, as here you can see here. So answer here is nitrosomonas, because here you can see nitrobacter, nitrospira, nitrococcus. In the previous question, the bacteria were here they were nitrosospira, nitrosococcus, but here so is missing. So here, the answer is nitrosomonas. A is the answer. Which of the following are aerobic bacteria? So we know nitrosomonas and nitrobacter, which are nitrifying bacteria causing nitrification in 2 steps, are aerobic bacteria. So A is the answer. Which of the following processes involves the simultaneous conversion of an amino acid into keto acid and a keto acid into amino acid? Okay. So what is the question? In which process amino acid becomes keto acid and a keto acid becomes amino acid? You have studied that process is called as transamination, because there is just transfer of amino group from amino acid to keto acid, isn't it? So that this interconversion is possible. B is the answer. In transamination, there is a transfer of an amino group from what? Okay. So you have studied transamination. You have studied the example, glutamic acid, your glutamic acid plus oxaloacetic acid, it gives rise to alpha ketoglutarate acid plus aspartic acid. Yeah, that is the equation of transamination, typical equation, which starts with glutamic acid, that is amino acid. And this glutamic acid is called as donor amino acid, which donates its amino group to keto acid, that is oxaloacetic acid, so that this keto acid becomes amino acid, product amino acid called as aspartic acid. And the your donor amino acid, it becomes your keto acid, that is called as alpha ketoglutarate acid. So here the answer is transfer of amino group from amino acid. Glutamic acid is amino acid which donates its amino group to keto acid called as oxaloacetic acid. So here the answer is option C. Okay, glutamate dehydrogenase is the enzyme which catalyzes what? Reductive amination of this or transamination of this. So you studied in transamination, the enzyme is transaminase. When the enzyme is dehydrogenase, it is the enzyme of reductive amination, in which your amino acid, your keto acid is reduced to form amino acid. So that is called as reductive amination, isn't it? Keto acid is reduced to form amino acid. For example, alpha ketoglutarate acid is the keto acid where, which when gets ammonia or ammonium, then it forms amino acid. And then this amino acid, in presence of enzyme glutamate dehydrogenase, it forms your glutamic acid or glutamate. So when we start with alpha ketoglutamic acid or alpha ketoglutarate, which is a keto acid, it forms amino acid in presence of your ammonia, and that then in presence of glutamate dehydrogenase gives rise to glutamic acid or glutamate. So B is the answer here. This is the equation of your reductive amination. Reductive amination. B is the answer. Which of the following is a group of denitrifying bacteria? So various bacteria are given and you have to identify which is denitrifying group. So A is the answer. Bacillus, Pseudomonas, and Thiobacillus. All these are species of denitrifying bacteria. Specifically, Thiobacillus denitrificans, Pseudomonas denitrificans, and Bacillus denitrificans. They cause denitrification, that is conversion of nitrate to nitrogen. So A is the answer here. Then which of the following process is considered as the reverse of nitrogen fixation? Exactly reverse of nitrogen fixation. So nitrogen fixation starts from nitrogen and it produces ammonia. And which is that process which gives rise to nitrogen again in the environment, which releases nitrogen back to the environment. And that process is called as denitrification, in which nitrogen is formed from reduction of nitrate. So exactly reverse of nitrogen fixation is called as denitrification. D is the answer here. Okay. Which of the following are true about the heterocyst, which are present in some filamentous cyanobacteria? Okay. So, well, it is a site of nitrogen fixation. Yes, that is right. Inside heterocyst, nitrogen fixation takes place. It consists of photosystem 1 only. That is also right. It has additional cell wall. It has very thick wall. That is also right. It provides micro anaerobic environment. And this is also right, because when photosystem 1 only is present, there is oxygen is not released, so that some anaerobic condition is, because the enzyme nitrogenase is very sensitive to oxygen, and here there is no provision for leg hemoglobin. So they have to provide some structure like this heterocyst, which will give you micro anaerobic condition for nitrogen fixation, for proper functioning of nitrogenase enzymes. So all of these are correct here. So when you see the option, you will get the clear picture of the correct answer. That is D is the correct answer. All of these. Okay, option D is right. Then which of the following reaction of nitrogen cycle constitutes the direct conversion of nitrite and ammonium into diatomic nitrogen and water. So see here, direct conversion of nitrite and ammonium into diatomic nitrogen and water. So we have studied that ammonia is oxidized to nitrite and then nitrate. Okay, and that is aerobic condition. But sometimes anaerobic conditions occur. Under anaerobic condition, this ammonium is oxidized, and that process is called as anammox. As you can see here, ana means anaerobic, ammo means ammonium, and ox means oxidation. Anaerobic ammonium oxidation. So this is anaerobic in the sense this ammonium is acted upon by some bacteria in absence of oxygen. Instead of oxygen, they take nitrite as the final electron acceptor, and then they get energy by this oxidation. And so here the answer is B, 40. Question number 40, answer is B, correct. Option is option B. Okay. So these are some of the references. Wagner, Bernard, biochemist, some of the websites I have I have consulted, and Ganguly Das, again, College Botany, good book. Then one book from Open University, and this is your textbook. You can use any textbook, no problem, for getting such a type of multiple choice questions. So if you have, thank you very much for kindly listening to this. If you again, if you have any problem, any query, any difficulty, then you can post it in the chat box, or you can ask me directly. My email ID is given in the very first slide, and also I have given my phone number also. If you feel very urgent, in that case only you can call me. And so study hard, study this again. All the best for your forthcoming examination. Thank you.